A very good morning, dear students. Today we are going to learn about the sugarcane diseases. It's a very important diseases because in every year sugarcane is being destroyed due to the attack of various plant pathogens. And we know plant pathology deals with the study, symptoms, management of the diseases, and mainly from India. As we are from India, India is each year a number of crops have been destroyed due to the attack of various pathogens. And sugarcane is a very important crop because a huge number of sugar has been utilized for our daily consumption. It is being exported also, and it is used in various sugar byproducts for making many sugar byproducts. So that is very interesting. So today the most important points will cover the contents. The contents will about see the introduction, systematic position, symptoms, the causal organism, disease cycle, control, conclusion, and definitely references. Because each and every study we should have some references. The most important references, most relevant references also help us to know the disease cycle, their occurrence, what is their present status is going on in India as well as in the abroad. So as in the first slide in the introduction, we saw the fungus known as Coleotrichum falciparum, which causes the red rot of sugarcane. It is a very serious disease and is first described by Wayne in the year 1893, because it was previously the pathogen is not being identified by any of the pathologists, but due to his vigorous work, Professor Wendt, he did a vigorous work on symptoms, studying the symptoms, isolation, and all these things. And for his great work, we could come to know about the disease, the causal organism, that is the Coleotrichum falciparum. Okay, the disease causes damage to the sugarcane, mainly reducing the sucrose content of the cane juice. We know the sugarcane is mainly popular for yielding the sugar juice, that is the sucrose. So if the pathogen, and that is the main reason for the pathogen to get attack the sugarcane, because it get lot of sugar. When the pathogen is getting lot of sugar content, then definitely the host will be much more susceptible for the attack of various pathogen. So in the sugarcane also, the pathogen attacks the sugarcane internode portion, and the internode portion they try to get all the sucrose for their nutrition, and slowly the main sugar content of the plant will reduce. And if the sugarcane is attacked by this type of fungi, definitely we are not going to get the satisfactory yield or satisfactory sugar content out of our cultivation. Okay, the pathogen mainly spread in the rainy season, because rainy season, we know rainy season is very favorable condition for the any plant pathogen to get attached with the host, because we know the minimum temperature 24 to 25 degrees centigrade is very important for any pathogen to get invade through the stomatal opening of the plants, mainly in the morning time. Okay, so that is spread. You can see the symptoms. You can see the symptoms, how the internode area being affected. So if we cut the sugarcane stem from the middle, you can see how the pathogen gets its path through the vascular bundles. You can see the all the mainly the vascular bundles is made up of they all be attacked. The germ tube, all the infection thread has been infected through the through the main region of the stem. Clear? So that's very interesting to know. So systematic position. So it is Eumycota, division Eumycota, within the modern fungi is all under common Eumycota, subdivision Deuteromycotina, class Coleomycetes, Melanocinils, family Melanconiaceae, Coleotrichum falciparum. Deuteromycotina, we know these are the imperfect fungi. We know it very well. They do not show any typical sexual or asexual reproduction in their life cycle. So it is very interesting how to know about the life cycle of the disease. Okay, so symptoms. Most importantly, so whenever we go for any type of fungal study or fungal pathogen identification, before that we have to know the symptoms, how the pathogen um, creates the symptom in the host plant, or the host parts. By seeing the symptoms, at least a plant pathologist, as a experienced plant pathologist, can give some idea about the pathogen, to show their various disease cycle. So red rot only be observed when it is being completely rotted in the interior and lost nature be a bright color. So in the beginning we cannot see it is being affected by the sugar cane. This is pathogen. Okay, so when the pathogen gets affected, when the pathogen is the host affected by the pathogen completely, then only the symptoms appear from the outer side, as you can see in the previous slide. See, okay, so that is very interesting. So in the symptoms we can see plants can be seen yellowing, shriveling and drying on the upper leaves. Okay, the disease can be recognized by the longitudinal reddening of the normally white or yellowish white in the internode area. Okay, in the internode area we can see normal reddening of the tissue. uh, Late in the season, minute velvety dark dot-like structure acervuli develop near the nose disease nerve. Acervuli is nothing but the fungus reproductive stage. You see, in the beginning, these symptoms appear on the leaves. So in the mature stage, all the internode area being get affected. You can see some blackening portion is being affected, and slowly this will also cover the internode, and the internode is not affected because internode is because the node area is a very strong area. So strong area, it cannot be affected by the fungi, but what they do, they produce the some spores. Okay, they produce the spore and they get involved in this area. Okay, so these are the affected area, and these nodes area actually they produce the spores which are in a spore sac. This is known as acervuli. This is a fungus spore structure, and they will present inside that. Okay, so these are the symptoms. Okay, then causal organism, already I have discussed, red rot of sugarcane caused by Colotrotrichum glomerula. There has been constant difference in the opinion as to be the natural fungus and causes of the disease. Some insisted that this fungus is mostly strictly saprophytic, and some used to say parasitic. Others say that it can attack mature canes except through the wounds, but it can be attack young plants also. However, the young canes are usually protected by the leaf sheaths. You know, the young plants, young sugarcane plant, if you see, is a very simple, it's a very, it's totally covered actually. Okay, but when it is being matured, the internode gets increase in size. When the internode is getting in size, then there is huge chance of attacking the pathogen. Okay, because pathogen knows it very well to get the space in the internode area, because their main target is to attack the internode area, to draw the nutrition out from the internode area. Okay, so that is very key point to study the sugarcane disease. Okay, then disease cycle. So disease cycle is very important. The sources primary inoculum are the old fragmented stalks, leaves which fungus grows saprophytically. Okay, according to the available evidence, seeds sets from disease canes are also the chief means of survival and annual reoccurrence of the disease. Suppose some seed-borne pathogen is also there. Colotrichum falcatum, the pathogen can be also associated with the seed and even with the disease portion. Suppose in the previous years the sugarcane disease occurs, and if it is not cleared, if the field is not washed or not burnt properly, then the fungal spores can be there as an inoculum, which may reinfect the crop in the next year. Understood? So that is a very uh, important point to know about this disease cycle. Some candidas are disseminated by winds, irrigation of water, like that. They could conidia actually germinate by germ tube, which on coming in contact with any hard substances, soil particles or plant parts, or in the form of appreciarium from which the infected hyphae is produced. We know Episerum is the main target area from where the germ tube develop, after which the fungus used to infect the whole plant. Okay, so in a very simply we can say that how to stop the disease. So disease stop as a, it's a very simple idea I can give to all of you, that we have to stop the we have to stop or burn the previous affected parts of the sugarcane leaves, and mostly we have to go through a very certified seeds. Okay, so that you should wear a certified seeds. We have to constantly go through, we have to constantly check our field conditions, whether the field is very much equipped with all the sanitary or the sanitation properly, is the sanitation is being done properly or not. Because if the field is not well sterilized, then it is definitely the fungus is going to attack. Okay, so in the, this is the cycle. You see how the, in the active phase, how the germination, then they go germination window, they produce by the germ tube, they enters to growing seed or seedling stage, then mycelium, just hyphae, slowly they develop into primary mycelium, and we know primary mycelium is developed into secondary mycelium, which is also known as the fruiting stage. Okay, these secondary mycelium then undergo infection. When the secondary mycelium develops inside the plant, then only the infection, host infection appears. Okay, then slowly they produce the mycelium, then conidia, and then slowly they will produce the chlamydospores. Okay, that is why it is being divided in the dormant phase and the active stage. In the dormant phase, the perennation occurs. Okay, the actually the spores get attached with the cell of the sugarcane, but in the active phase they grow, they produce the symptoms. So we have to wait until the active phase is being shown in the our field. Okay, it may take some time from to show the dormant phase to active phase. It depends on the environmental condition, environmental humidity. If the favorable environmental is there, definitely the dormant phases can transform into active phase, and then only we can see the symptoms on the leaves, and slowly we can see this, so the symptoms in the cane, in the canes itself. Okay, so this is the simply disease cycle of the sugarcane. Okay, so control. So any type of plant diseases cannot be studied without their management, because nowadays we can come to know about the various diseases, various symptoms, various causal organisms, various diseases. But unless until we cannot give a clear idea about the management, how the farmers can protect their field, because they are not a plant pathologists like us, they are not a botany students like us, but we have to make them very clear and how you can do these things. Okay, so management is very important, and we have to give a clear idea of management to them, so that they can practice this very within a very limited cost. Okay, because they cannot afford in some areas, some field the farmers cannot afford so much of money for the management. So we have to go to the cultural methods. Okay, in the cultural methods we can do by disease free canes, we can use the disease free canes. 2-3 year rotation should be followed. Okay, grow resistant varieties, these are some resistant varieties you see. This is very important. Resistant variety has been developed by Agriculture Institute of Pusa, and Pusa is the New Delhi in all throughout the India. They are developing the developed varieties, and these developed varieties are disease resistant variety, and where the fungi cannot attack. These special varieties has some genetic advancement through which they they can develop the defense mechanism against the attack of the this Colletotrichum fulcatum pathogen. Understood? So that is why removal of affected clump and soil drainage is very important by the carbamazepine gene. By 50 W, we can cut the endos entire set should be deep in a fungicide solution. Okay, in body X mixture. That's most importantly, we have to follow the concentration. If the concentration is more, it can affect the it can go into the tissue of the sugarcane, which will be consumed by us, and it could be a major cause of our health hazards. Okay, so every time for any for using of any type of fungicide, we have to be very much clear that that concentration has to be maintained. If we increase the concentration, so to control the particular pathogen, it may directly affects our food chain. Understood? So that is very interesting point. We have to always make it very clear about the concentration when we are using any type of fungicides. Okay, we can do some low lying areas avoid for cultivation. Disease plant should be dug out and burned. Okay, so these are some control measurement. We can do heat therapy, hot water treatment we can do for 52 degree centigrade for 18 minutes. Steam treatment we can do for 4 to 5 hours. Hot air treatment for 2 hours. So that is very important. Heat treatment, what happens? Mostly the conidia used to disintegrate disintegrate by the attack of the heat. So in the normal heat, if we can maintain the temperature and the time, definitely we can protect the sugarcane from the attack of this pathogen. Okay, some chemical methods is very important also, where we can we see the carbonate dazium. Before planting, we can spread some carbondazim in a minimum concentration. Use fungitoxic chemicals like bevistine. Bevistine is a very well uh, used fungicide in many crops from the attack of various pathogen. Uh, so it is we can use for 0C, only 0.1% we have to use. 0.1% for 18 minutes at 52 degrees centigrade, or dipping whole sets in a complete elimination of the disease. Okay, so slowly the management is very important. So in conclusion, we say is this is the end of our presentation, but we'd like to thank all your time sincerely that we had a pleasure to present before you all. So it is very easy to understand the various diseases in crops, but we should know about the disease cycle, about the management, and about the nature of the pathogen. So that is why today, in a very very concisely, I have discussed about the red rot of sugarcane, which is by the pathogen Colletotrichum falciparum, and this is under the group of Deuteromycotina, and we have seen various symptoms. Okay, symptoms, causal organism, management, disease cycle. I hope it will definitely help you for your preparation of your final studies. It definitely help you in your future preparation. Thank you.